All right, welcome to the pre-lab video for the kinetics lab. So in this lab, we are going to study the reaction kinetics of the enzyme carboxyl esterase. This enzyme catalyzes a number of different reactions, but the reaction that we are going to examine during the lab is p-nitrophenyl acetate plus water. Water is being used for hydrolysis. And the products are p-nitrophenol and acetate. So during the different parts of this experiment, you are going to study the effects of a number of different variables on the reaction rate overall. So first you'll look at the effect of enzyme concentration, the effect of substrate concentration, and as we have already talked about during the lecture portion of this class, both of these aspects can affect the rate of a reaction. We're also going to look at the effect of butyraldehyde, which is a competitive inhibitor. As we have also learned, competitive inhibitors will compete with the substrate for access to the active site. And as you should also know, this is going to probably slow down the reaction rate. And then finally, we'll look at the effect of inactivation of carboxylase using a molecule that will inactivate the enzyme. So it's different than a competitive inhibitor. Our inactivator actually modifies the enzyme in such a way that it's no longer able to carry out the reaction or to help catalyze the reaction. I should also mention that this reaction that we're monitoring is spontaneous, but without an enzyme, it's very, very slow. So carboxyl esterase is really important for helping to make this reaction happen at a sort of reasonable rate that's useful in our bodies and in our cells. So let's look at the different reagents that we're going to use during our experiment. So first we'll use a buffer. We're going to use for our buffer tris, and this is to help keep everything at a constant pH of 8. We are also going to use our enzyme, which is carboxyl esterase, which is going to catalyze the reaction that we're monitoring. We also have our substrate, which is p-nitrophenyl acetate, our competitive inhibitor, which is butyraldehyde, and our inactivator, which is bis-p-nitrophenyl phosphate. And I just want to note that we'll have a number of different solutions that contain various concentrations of some of these. There might also be solutions that have different combinations of some of these different reagents within them. So please be careful to read all of the labels very carefully and make sure that what you're intending to add for each reaction that you're monitoring is what you are actually adding, especially because some of these components have very similar names to each other. So I encourage you to be very cautious and careful as you prepare all the different samples that you're going to analyze in this experiment. So that brings us to actually running the reaction. So what you will do is combine the reagents together in a cuvette. You're going to add buffer and substrate and possibly some other components such as the inhibitor and the inactivator, and also, of course, the enzyme, which helps to catalyze the reaction and make it happen faster. But as you can see, enzyme is written here in red. It's really important that you add the enzyme right before you start the measurement, because as soon as you add the enzyme to the cuvette, the reaction will begin. And it's really important to measure the reaction rate at the beginning of the reaction. You want the initial reaction rate. So when you combine all the reagents together, add the enzyme last, then mix thoroughly but quickly, and then get it into the spectrophotometer as soon as possible to start measuring. That brings us to the next point, which is that we're going to measure the absorbance at 405 nanometers for 1 minute. And you can see there are pictures here of the two different spectrophotometers we have in the lab. So over here we have the Curie, down on the bottom we have the Vernier. They're both capable of a kinetic measurement. So we'll try to get them all running for all of you so you can all be able to do the experiment at the same time and not be waiting for a long time. So overall, this assay should sound pretty familiar because it's very similar to the LDH activity assay that we did with a few key differences. So first of all, we're monitoring the accumulation of a product instead of the loss over time of the reactant, the substrate. This time we're measuring a visible color change. Last time during LDH, we were looking in the UV spectrum. We're also going to expect to see a lot less noise in our data. Some of the LDH activity assay data was relatively sort of messy looking. There was some of the measurements had a lot of noise in them. Here we should be able to see some pretty nice clear curves. Another major difference here is that for the LDH activity assay, we didn't know how much enzyme we had, and we did the assay to sort of figure out how much enzyme there was. In this case, we're going to be adding known amounts of all of our different reagents and components, and just be looking at the effects of varying all of those different components, including substrate concentration, enzyme concentration, inhibitor, and inactivator. So that brings us to what your reaction kinetic curves might actually look like. So generally what we'll expect to see is a fairly linear reaction kinetic that has a positive slope. Like I mentioned, we're looking for the accumulation of a product, and that product absorbs at 405 nanometers. So over time, as the enzyme converts more substrate into product, you will see more product forming. That being said, you might also see a curve that looks a little more like a curve, where there's an initial very steep reaction rate that slows down over time, and this is just because the product, or excuse me, the substrate you've added to the cuvette is being consumed and being turned into product, and so as you use up the available substrate in the cuvette, less and less of it can be converted, and so the reaction is going to slow down because there's less substrate available. This is also perfectly fine if you get this sort of data. Just keep in mind that what we're looking to measure is the initial rate of the reaction. So you'll want to try to find the linear portion, the closest to your initial time, your time zero, and just use the linear portion of your curve when you're calculating the actual reaction rate. Because as you know, the reaction rate is going to be the change in absorbance over time, or the slope of this curve. So when you have just a line, as we see on the left, it's very easy to calculate the slope. When you have more of a curve, like we see on the right, you'll have to find essentially the linear portion of your curve to help you find the slope of that initial reaction rate. Depending on which reagents you have in your cuvette at any given time, you might have lines that have different slopes. And as you know, the slope is telling you the reaction rate. So this green line that we just added on the left has a higher slope than that red line. So that is measuring a faster reaction rate. These two lines, though, they do have the same y-intercept. Sometimes you might see, or during some of your samples that you're measuring, you might see a line that has a different y-intercept than some of your other ones. This is also perfectly fine. As I've been saying, it's really what we're measuring here is the slope of each line. The y-intercept is less relevant. It's really more of a measurement of how much initial substrate and product you have in that cuvette. It's not measuring the rate of the reaction. Again, the rate of the reaction is the slope of the line. So in this case, we have on the left the red, the green, and the blue lines. The green line of all of those three still has the steepest slope, and so that green line represents the fastest reaction rate. So overall, what you should expect to see during this lab are these four different components that we're measuring. First, the effect of enzyme concentration. When you add more enzyme, you should expect a faster reaction rate because there's more enzyme there in your cuvette catalyzing the reaction. Similarly, when we look at the effect of substrate concentration, you would expect to see that as you add more substrate, you'll have a faster reaction rate because the more substrate you add, the closer the enzyme will get to being saturated, and so the higher the possible rate can be. When we're looking at the effect of butyraldehyde, this is our competitive inhibitor. The competitive inhibitor is competing with the substrate for access to the active site in our carboxylesterase enzyme. So the competitive inhibitor is going to slow down the reaction rate in comparison to when there's no inhibitor there, because there's less formation of the enzyme substrate complex. So that brings us to our last component that we're measuring, which is the inactivation of carboxylesterase. So the more time the inactivator spends with the enzyme, the slower the reaction rate will be because as the inactivator interacts with the enzyme is inactivated. So it cannot catalyze reaction, and so it's essentially like you've taken away potential enzymes, so you get a slower possible reaction rate. Something interesting also that you will be measuring is combining together the inactivator with the competitive inhibitor. So the competitive inhibitor, butyraldehyde, we mentioned how it's competitive with the substrate for access to the active site of the enzyme. It's also competitive with the inactivator. So if you add both butyraldehyde, the competitive inhibitor, and our inactivator, you'll actually see a slowing of the rate because the inactivator has less access to the enzyme because it has to compete with that competitive inhibitor. So the rate will slow down, but it will slow down more slowly than it did when there wasn't also a competitive inhibitor in there. So that brings us to the analysis that you're going to do after you complete this lab. So what you'll do is you'll find the slope of each reaction kinetic. For the curves that are not linear, you can just find the initial slope, that most linear portion early on in the reaction. And you can use the equation for a line to help determine the slope, which will give you the rate of each reaction. And then once you have that information, you'll be able to calculate the initial rate of the reaction using Beer's law, which is written here. Absorbance equals extinction coefficient times concentration times path length. And then finally, you can graph the rate as a function of enzyme concentration, substrate concentration, and inactivation time. So when we looked at our reaction kinetics, we were measuring the absorbance as a function of time. But now we're going to be measuring the reaction rate as a function of the concentration of our enzyme and substrate, as well as time spent with the inactivator. And so on these types of plots, so for an example, we have our rate as a function of enzyme concentration. So for each enzyme concentration, you'll be calculating a rate. And so presumably, as the enzyme concentration increased, your initial rate also increased. So you'll probably end up with something that looks a little bit like this, where for each time your enzyme concentration increased, you also saw an increase in the rate of the reaction. And you'll think more about this analysis after you complete the lab and as you look at your instructions that are in the lab report instructions document. So finally, after you make all of those graphs, you'll be able to compare the trends that you see to the expected results that we just talked about in the previous slide. And now you should be ready to complete this lab and answer the pre-lab questions and come prepared to the lab on Wednesday morning.